So my name is Russell Engelman, and today I'm going to be presenting to you on body size estimation and body shape reconstruction in Dunkleosteus terrelli. So in this talk, I'm going to cover several things. What is Dunkleosteus terrelli? Where is it from? The Cleveland Shale. How big was Dunkleosteus? What is new research saying about the body size and shape of Dunkleosteus? And what does this mean for overall patterns of vertebrate evolution? So Dunkleosteus is one of the most widely recognized prehistoric animals. And it's recognized because it's considered to be one of the first large megafaunal vertebrate apex predators. Dunkleosteus itself is named for David Dunkel. The name means Dunkel's bone. And David Dunkel was the curator of vertebrate paleontology at the Cleveland Museum of Natural History between 1939 and 1946. So Dunkleosteus is a type of fish known as the Arthrodira. Arthrodires are part of a larger group known as Placoderms. And this is a group defined by several features, and this includes armor plating on the head and torso. They have both paired pelvic and pectoral fins, whereas earlier fishes generally only had a pair of pectoral fins. They have jaws but no teeth, kind of. That's something that it's a bit too complicated to get into here. And this group has a very short fossil record of only about 60 million years. So the relationships among placoderms within invertebrates more broadly are very controversial. And it's largely due to things like this. This is Engelman-PhD primordialis. This is one of several forms that are known as maxillate placoderms. These are they're mostly coming out of China. And in phylogenies, what's happening is when you put these things in phylogenies, instead of getting a monophyletic single branch of placoderms, instead these placoderms appear to represent 5 or 6 different lineages at the base of the broader jawed vertebrate family tree, with several groups being progressively more closely related to modern jawed vertebrates like bony fishes and sharks. The maxillate placoderms are thought to be closely related to modern fishes. Arthrodires are thought to be more closely related to modern fishes than, say, the antiarchs, which are way up at the top right corner. And as you can see, it's kind of this series of branches diverging from the early jawed fish ancestor. So Dunkleosteus is from the Cleveland Shale. And this is a black shale unit in northeastern Ohio. It's a black shale because the rocks contain very high amounts of organic hydrocarbons. And this rock layer dates to the late Devonian period, between 363 and 360 million years ago. And this is significantly older than the dinosaurs, just for reference. So during the late Devonian, the Cleveland area was actually part of this very narrow inlet of the ocean called the Kaskaskia Sea. The way to think about this is it's kind of like the Chesapeake Bay today. It's this very long, narrow inlet incising into the continent from the outside. This would be a very narrow inlet that's bordered on the west by the broad North American continent, and then on the east by the rising Appalachian Mountains. And this sea was very unusual because in the offshore waters, you had this arrangement where the surface layers were heavily oxygenated and had high productivity, but then the lower layers were heavily anoxic. There was no oxygen in the water and nothing could survive there. And that's what actually produces these black shales. And what this means is that basically nothing can live on the bottom. So when you get these ecosystems, they're essentially a pelagic ecosystem in isolation. There is almost no influence from the coastline, and there's almost no bottom dwelling or super deep sea life that you'd expect from modern ocean. So it's a very unusual looking ecosystem when you start considering it. So the Cleveland Shale is best known for its fossil fish fauna, and there are about 60 species known from this general area. About half of these are sharks. The Cleveland Shale is very well known for its shark fauna, and specifically, their sharks are well known because when they are preserved, they often show complete body outlines with soft tissue and organs, and that's very, very rare in the fossil record. There's also 5 species of bony fishes. There's very little to say about them. One of them is part of the base of the food chain in some ways, but the other ones are they barely show up. They're very rare components of the fauna. And then the other half of the fauna is mostly composed of arthrodires. And these are, as mentioned, this group of fishes that is very heavily characterized by the front half of the body having an extensive set of dermal plating over it. This is the group that includes Dunkleosteus, and this is the group that we're mostly going to be talking about for the rest of the lecture. So at the very top of the Cleveland Shale correlates with what's known as the Hongenberg event. They've actually done research in over by Tiedemann Road down by the airport, and they found a chemical excursion signature at the very top of the Cleveland Shale that matches the chemical excursion of the type section of the Hongenberg event over in Germany. And what this means is that at that section, something very, very bad happened at the top of the Cleveland Shale. You may have seen lots of dinosaur documentaries where the paleontologist uses with a cowboy hat goes out to North Dakota and points at this little whitish band layer and says, this is the layer where the dinosaurs died, this is the layer where they find the iridium where the asteroid impacted. We actually have a very similar layer in Cleveland, but it's only detectable by geochemical analysis as opposed to just looking at it. So the Hongenberg event is the last stage in end-Devonian extinction. The end-Devonian extinction is kind of complicated. I don't really want to go into what exactly. So there's really, I don't just say the end-Devonian extinction. There's multiple parts to it. It's a very strange extinction. And in total, the end-Devonian extinction is considered one of Earth's big five mass extinctions. About half of all life on Earth is thought to have gone extinct over this broader period. And at this point, you're probably wondering what caused the end-Devonian extinction. And what's increasingly looked like is the most likely culprit is that trees murdered everyone. So basically, the thought is that when trees evolved for the first time, the sudden increase in terrestrial biomass drew down a huge amount of oxygen from the atmosphere and increased terrestrial weathering. So you had massive amounts of eutrophication into the oceans, which they hadn't really handled this before. And on top of that, the mass drawdown of CO2 into organic tissues probably caused a snap ice age. We have evidence for this. We have evidence for this from several areas. This is probably the most influential of this. This is something called the Robinson Dropstone. This is a glacial dropstone in Kentucky. The problem is that it's not a Pleistocene one. It's actually embedded in the Cleveland Shale. It's also a bit too far south for any of the known glacial events. And they've actually been able to link it to a parent rock unit somewhere in the Appalachians. So this is a glacial dropstone that was deposited about the same time the Dunkleosteus was swimming around. The problem is at this time, Cleveland was approximately the same latitude as the Tropic of Capricorn, with the same latitude as Rio de Janeiro. Like, you do not get glaciers at Rio de Janeiro unless something very wrong has gone with the environment. So Cleveland Shale fossils have been collected for nearly 150 years, and it's actually very difficult to collect in the broader Ohio Shale, and so collection tends to proceed in very major spurts. Again, the problem mostly is that there's trees everywhere. The fossil sites in the American West don't have this problem, but fossil sites in the East always have this problem with trees and plant cover. So in the 1860s, there was the first initial spurt of collections. It was mostly introduced by the introduction of blasting equipment into civilian construction post-Civil War. And this is when fossils first started to be collected along the Lake Erie coastlines. And this led to, eventually, this big monograph of the Ohio Geological Survey that was authored by John Newberry. And this is actually the work that originally named the species for Dunkleosteus, but not the genus. That's something I don't have time to go into. So the next big period of collection was in the 1920s, associated with the Cleveland construction boom. I don't know if anyone here knows this, but Cleveland at one point was a very major American city, and it was growing very fast. And mostly this was the 1920s. So as people were constructing and bulldozing everything, they kept running into fish bones. This is when collections started to move away from the coastlines into like the river valleys, like the Rocky River. And this is actually the period in which the Cleveland Museum of Natural History collected most of its specimens, including the big famous Dunkleosteus head you see in their collections. And then finally, in the 1960s, we have the construction of I-71. When the interstate system was built, they had to basically bulldoze a swath straight through Cleveland, and as a result, they exposed a lot of fish bones. And what the Cleveland Museum did is they would basically walk behind the bulldozers, note every single scrap of fish bone they could as it was being exposed, and then would collect it as fast as possible. It was basically a salvage operation for this stuff that was going to get destroyed. And in fact, a lot of stuff was destroyed. They did the best they could, but, you know, there was a huge swath of rock being exposed, and there were only so many people. They organized this huge effort, and this actually got quite a bit of national attention in terms of what was being done. So the thing is that research interest on Cleveland Shale fossils basically dropped off the face of the earth after 1930. There is very little research that was done between about 1947 and 1991. There was basically nothing going on. I often point to this manuscript, 1932, which was Heinz's monograph of Dinichthyes, which it's Dunkleosteus. That's the old name for Dinichthyes terrelli is the old name for Dunkleosteus terrelli. And that was kind of one of the last major landmarks in Cleveland Shale paleontology. As a result, most of the fossils that have been collected from the Cleveland Shale have never actually been described. And this includes pretty much all the specimens of Cleveland Museum of Natural History. This actually includes the big famous head, You've seen all over the place. This thing has never actually been described. There's no paper describing it. You cannot find it in the literature. And I actually know the reason for this. So as I mentioned before, the last big monograph on Dunkleosteus' anatomy was by Anatol Heinz in 1932. Heinz knew about the material from Cleveland because it had been exhibited this time. He explicitly did not include any specimens from the Cleveland Museum in his work. And he says this. He says this explicitly in his work. And the reason for this is he did not want to scoop the work of his colleague, Jesse Hyde, who he said let him in the museum to see all this stuff that was under description. And Hyde said he was working on a monograph series of all the stuff that Cleveland Museum collected. Hyde was also a perfectionist, and he ended up he did a lot of things for the museum. Like he organized a lot of his collection efforts, but he never wrote anything up. And he would delay publishing year after year because he wanted everything to be perfect. Eventually, his health caught up with him, and he ended up never publishing anything on the Cleveland shale. So this has resulted in the Cleveland shale being forgotten about, even in the very, very niche specialist world of Paleozoic fish workers. These are two papers that are written about 30 years apart. This is Miles 1969. This is Carr 1995. The papers are about 30 years apart, and their wording of the Cleveland shale lamenting the lack of knowledge about these fish is almost identical. Or let's get a more recent example. These are two review articles on the history of fossil fish and advances in fossil fish research in the last 20 or so years. These sentences are literally the only contribution the material from Cleveland has made to our understanding of fish evolution. It basically doesn't exist in the literature, and it's a brief mention of, ooh, look, big scary monster, let's move on to something else. Again, these are forgotten about even in the very niche world of Paleozoic fish workers. They're not very well understood. And that's the problem. So Dunkleosteus is well known. This animal is probably one of the most popular prehistoric animals. It appears in all sorts of popular media. There's figurines made of it. There's merchandise. But the animal actually isn't very well studied. And so as a result, we actually know very little about what this thing was like as an animal rather than some movie monster. And so when people ask paleontologists about very basic information about this species, like how long was it, what did it look like, where did it live, how did it live, what was its life cycle like, most of the time paleontologists just shrug their shoulders and say, well, we don't know. And this is actually a broader problem with the Cleveland shale in general. The state of knowledge about the ecosystem of the Cleveland shale is kind of similar to the state of knowledge about like Mesozoic ecosystems in the 1950s. Now this kind of reflects a trope that paleobotanist Kurt Johnson called dinosaurs in parking lots. This old idea of the Tyrannosaurus rex or some of the dinosaurs stomping around a desolate badlands. There's no plants. There's no other parts of the ecosystem, completely divorced from any natural history, ecological context, with volcanoes going off in the background. And we're basically about the same position with Dunkleosteus and the Cleveland shale. It's completely divorced from what this animal actually was. And this goes for anything from the Cleveland shale. Dunkleosteus is actually one of the two best-known things from the Cleveland shale, and everything else is worse off. So a lot of this is because the post-Jurassic anatomy of Dunkleosteus is very poorly known. So Dunkleosteus has a bony head that also includes the anterior half of the trunk, but then the rest of the body is composed of paraconch ossified cartilage that very rarely preserves. So again, yeah, only the head and thoracic arm are actually preserved in the fossil record. The rest of the body basically rots away, and this makes it very difficult to say anything about body size or shape. So if you go back and look at old literature, generally lengths of greater than 5 meters are traditionally suggested for Dunkleosteus terrelli. But the thing is that these estimates are almost entirely speculative and based on what is effectively no data. They don't say how they calculate these estimates. They don't say what specimens they measured. They don't say what comparative specimens they measured. They just say, we think Dunkleosteus was 6 meters long, and what's the evidence? Trust me, bro. And it's the problem because body size affects almost every part of an animal's biology. Body size affects dietary habits. You can only eat things that are so big relative to your body size. It affects ecology. Like, for example, you don't see many elephants climbing trees. That's a factor of size. It affects life cycle. Bigger animals reproduce more slowly. It affects comparative anatomy. Like, let's say you have a jaw. Does this animal have a big jaw relative to its body size or a small jaw relative to its body size? You need body size to figure that out. There's been a number of actually very good publications that talk about this. There's this famous quote by G.A. Bartholomew that is often brought up in this context that people like to cite all the place. That's very true. I would recommend Knut Smith-Nielsen's book, Scaling, about why this is important. It's a very good book for an introductory person who knows very little about biology or math to tell them why, give good explanations as to why this is a big issue. And this is something that has actually affected previous studies of Devonian research. A lot of people have run into issues where they need the size of Dunkleosteus or its relatives to do some kind of paleobiological analysis, and as a result, they end up either tiptoeing around the question or they use some kind of speculative size estimate. So I ended up doing some research on cleavage shellfishes, and I at first attempted to use traditional size methods for estimating the size of Dunkleosteus in my research. And the problem is they gave me biologically impossible numbers. And other researchers have, after I published my research, told me that they had similar issues. So, as I mentioned before, most of those previous estimates have no way of being evaluated. Like, there's no way to even test if they were remotely accurate. The most rigorous attempt to estimate size is Farrell 2017, who also noted the similar problem of most of these size estimates were basically just guesstimates. And Farron et al. Tried to provide a more rigorous estimate of estimating size by using upper jaw primitive in sharks. The assumption was that mouth size is correlated to body size, and therefore, if we have the mouth size of a big shark, we can just apply to Dunkleosteus and get a relative estimate of size. And they estimated lengths of between 6.9 to 8.8 meters for adult specimens of Dunkleosteus terrelli. The problem is that they never actually tested if their methods actually worked. Basically, they assumed that shark and Arthrodira mouth proportions were similar, but they never actually tested to see if the results they got were reasonable or like cross-reference them to see if, okay, this is a reasonable enough estimate of body size to go forward. They just assumed it did and never checked their assumptions. So we don't have a complete specimen of Dunkleosteus terrelli, but we do have complete specimens of these smaller Arthrodires, which we have basic complete body fossils for. And my first thought was, well, let's just check it against this and see if Farron's methods accurately predict the length of these things, because they're also Arthrodires. And it doesn't. So, as it turns out, mouth proportions of sharks consistently overestimate the body length of these complete Arthrodires. And this is the case whether you use upper jaw perimeter, like Farron et al. Did, or if you just use mouth width, which is a measurement that's a little bit more readily available in the literature for things like dogfish. So it's a little bit, I can add more dogfish in there to make the comparison a little bit better for smaller animals. And the problem is that this method, or either any method using mouth dimensions, tends to overestimate the size of complete Arthrodires by over 2.5 times the actual length. So Cacosteus here is about maybe 37 centimeters long. According to mouth proportions, it should be over a meter long, which is no, that animal's not that big. So Arthrodires basically just have wider mouths than sharks at similar body sizes. And in fact, if you roughly plot the line that you see for Arthrodires and you just stick Dunkleosteus in there without any second thought, you get a length of about 3 meters if you're just looking at Arthrodire mouth proportions. What this means is that these previous methods that use mouth length basically can't be applied. And the reason for this is that Arthrodires just have much larger mouths relative to body size. And Dunkleosteus in particular has this very elongate jaw with a slung back jaw, and it's a lot more similar to something like a viperfish than it is to a shark. And this is going to make any estimate based on mouth size or jaw size a massive overestimate. But however, the mouth size of Dunkleosteus, and I'm kind of skipping ahead just for 1 second here, relative to some of these later things that you're going to see, the mouth size of Dunkleosteus actually isn't that much different from other Paleozoic fishes. It's about 16% in very large specimens of estimated total length. And this is very similar to something like Chirolepis, this is an early bony fish, or Dracopristis, this is an early shark. So it's not that unusual in context. A lot of these Paleozoic fishes just have much, much bigger mouths than anything swimming around the oceans today. Not complete outliers, but like consistently you see larger mouths in these Paleozoic forms. Another way we can cross-reference this method of estimating body size of Dunkleosteus is we can use head length. And I'd also like to address a common misconception here. A lot of people think that this entire bony preserved region of Dunkleosteus is the head, and it's not. We know this because we know from anatomy where the brain case goes and where the gill chamber is. The brain case has a posterior extension that makes it kind of go over the gill chamber, but the thinky bits are in front. It's mostly comparable to other fishes. So basically, that's the head. These are the shoulder girdles because many of these bones are homologous with shoulder bone elements in modern fishes. And then this thing down here called the ventral shield is basically covers the sternum and belly region down to the level of the pelvic fins and the anus. So when we take the head proportions of Dunkleosteus and we assume the old links are correct, we end up with an animal with a significantly smaller head relative to body length than almost any other fish. This is significantly smaller than eels, I should point out. Like, that's eels, and the large specimens of Dunkleosteus are plotting at having heads that are only 8% of total length, which is not viable. The only things that these old estimates of Dunkleosteus would end up having heads similar to would be electric eels and oarfish. And if you know what an oarfish is, you can immediately tell that that's not a viable assumption for Dunkleosteus' body shape. So notably, these stars are other arthrodires, and other arthrodires show similar head to body proportions compared to other fishes. So we really shouldn't assume that placoderms just have these little tiny heads relative to body size. They just have regular heads relative to body size like any other fish. And even this thing, this is Amazictes, this is the closest relative to Dunkleosteus we have a complete body for. It does have a smaller head relative to body size, but it's an animal that's been considered ecologically similar to a Spanish mackerel or mahi-mahi, and its head-body proportions are pretty similar to that. So the thing is, yes, arthrodires have some variation in head proportions, but they vary in the same ways due to ecology as modern fishes. So we can use that to help inform exactly what we're doing. So when we're looking at why we can't have a Dunkleosteus that's 5 more meters long, this is why. This is what happens if you try to reconstruct Dunkleosteus using links proposed by prior authors. You get this. So it's very clear that there's been some kind of lapsus present. This is actually very clear because a lot of reconstructions of Dunkleosteus don't actually match the size of their own fossils. Like Ferrer's reconstruction is actually twice the size of the actual fossils if you scale it to the known measurements and you assume that it's supposed to be the length they claim it is. But what this means, it's very clear that we need a new method to estimate the body length of Dunkleosteus terrelli. So we need a new method, and this method must do the following. It must predict length in fishes independent of phylogeny, because we're estimating something very deep in the tree and we don't have a good phylogeny to use as a scaffold. It must include fishes that span the potential size range of Dunkleosteus to avoid extrapolation error. It must accurately estimate the length in other arthrodires like Acosteus, because if it doesn't do that, what's the chance that it works on Dunkleosteus? And then it must be measurable on the very limited skeletal remains we have of Dunkleosteus. This is a very tall order, but it turns out this is actually something we can accomplish. So one potential body size for arthrodires is what's known as orbitopercular length. This is basically just head length minus snout length. Head length has a very consistent definition in ichthyology. It's literally just tip of the snout to the end of the gill chamber. And basically, it's just omitting the preorbital portion of the snout. And the reason for this is that arthrodires have systematically smaller snouts than other groups of fishes. And if you try using the total head length to estimate body size, you end up systematically underestimating the size of arthrodires. So this is more fair to Dunkleosteus and gives a more accurate and somewhat larger estimate overall. And the reason this is expected to be a very good correlative body size is it extends across 2 regions, the neurocranium and gills, which are expected to be very highly conserved in fishes. When you estimate body size, you want to find what's effectively a load-bearing structure, something that cannot be modified too much relative to body size or else the animal's inviable. And the gills are a good example of this. If the gills are too small, the animal instantly suffocates and dies. If the gills are too large, that's a huge, expensive structure that natural selection is going to work to minimize as fast as possible. And perhaps most importantly, this is a measurement that is actually measurable in the remains we have Dunkleosteus. So the end of the head in placoderms is equivalent to the craniothoracic joint, which is the joint at the back of the head, and it matches up pretty well with the end of the gill chamber. The meaning of this can be summed up very simply. In general, short-bodied fishes have short heads, and then long-bodied fishes have long heads. The aspect ratio of both tends to be very highly correlated. There's been a couple of studies that actually suggest this. And what suggests is that there is something being conserved, like head shape and body shape in fishes seem to correlate in some ways, and that's a bit strange because we're used to land-living animals, but it does seem to be going on in fishes. And Dunkleosteus actually has a very short and deep head compared to the fishes, which suggests probably this thing is a relatively deep-bodied animal. So what I did is I collected a bunch of measurements of fishes, both in person from fluid-preserved specimens and from the previously preserved literature. And these fishes span a very, very wide range of taxonomic groups. And the idea is this is enough taxonomic breadth that we're probably going to be appropriately bracketing Dunkleosteus in this regression equation. And I also made sure to collect a very wide variety of body shapes. I collected eel-like fishes, very elongate torso fishes, fusiform fishes, and then there's even stuff like piranhas and butterfly fish in there just for the sake of it, just to see what happens. And then I took these methods and I applied them to complete specimens of Dunkleosteus terrelli that were housed at the Cleveland Museum of Natural History. So what I got is that OL very strongly correlates with total length in fishes. The R-squared for this regression model is about 0.95, and a single line accurately predicts body length across all fishes. The average error in this sample is about 17.6% of whatever the actual length of the fish is. So if you have this measurement, you will be able to get a value that's usually within plus or minus 17% of the actual value, which is fairly good. So when we add complete arthrodires to the mix, these are all these complete arthrodires we have for, they actually plot along the same line as sharks and bony fishes and everything else. So this seems to be a conserved proportion across all jawed fishes, and probably also lampreys for some reason. When you get to the Paleozoic jawless fishes, it's a little bit more sketchy. So orbital peritoneal length accurately predicts length in complete arthrodires. It predicts length pretty well in all of these. The only one it doesn't is Placodermi, and that one is because the specimen is damaged, and I actually suspect it's missing about 5 or 6 centimeters of it. What's interesting is that OL almost invariably overestimates body length. It usually doesn't underestimate it. And the resulting estimates are in plus or minus 12.8% of the actual length. And this is mostly because there aren't any eel-like arthrodires. There's no arthrodire equivalent of a butterfly fish. So they tend to be all fusiform to some degree. They're just general fish-shaped fish. So the model tends to work a bit better for them. So now we're going to pass Dunkleosteus terrelli, and we're going to specifically focus on this specimen. This is CMH5768, also known as that one you see everywhere. This is the big head they have at the Cleveland Museum of Natural History, and there are numerous casts of this specimen scattered all throughout the world. There's probably 30 to 40 casts of this scattered all throughout the world. Most major museums have one, and the Royal Ontario Museum actually has 2 for some reason. So when we apply this model to Dunkleosteus terrelli, we get much shorter lengths than any previous analysis. OL produces an estimated length of about 3.5 meters or 11.5 feet. And the question is, okay, given this is such a difference from previous estimates, is this actually reliable? So what I did is I ran a bunch of different analyses controlling for a bunch of different factors. Like I'd include all fishes, I would exclude discordant eel-like fishes, I would only include oceanic fishes, I would only include sharks, I would not include bony fish. I did tons of these, and they all came out the exact same. There's like 20 of these I did for the original study of this. I'm only showing like the most relevant here. None, like the highest any of them got was like 3.7, and a lot of them got closer to 3. Like using sharks only because arthrodires can be just, they slap shark anatomy on without considering anything further, you get 3 meters, which is even shorter than this. And so with any model there is, so the model isn't just this is how long it is probably this long, but it could be from yay big to yay big. But the thing to note is that the range of error in this model is not very large. Like we're not talking about error on the scale of plus or minus several meters. We're talking error on the scale of about 40 centimeters, which is not that big. And yeah, you could do some things different. Like you could twist, like the arm is a bit crushed. You could twist the arm around to make it a bit more streamlined. You could maybe assume it's a bit towards the upper end of the size estimate. But the thing is, like, even if you're assuming things, if you're assuming the error is underestimated slightly and it's a little bit higher than what this model is suggesting, you're not getting a 5-6 meter fish. You're getting a 3.5 to 4 meter fish, no matter what you do. In fact, these body proportions actually result in Dunkleosteus agreeing with other placoderms much more. So you can see here, that's the old estimates of Farrell 2017, and the new estimates actually result in Dunkleosteus being within the range of variation of non-elongate fishes, but just on the edge. The head is still unusually small relative to body size, even under this model. So now that we have estimates of length and we have body depth and width from fossils, we can create a new body reconstruction of Dunkleosteus terrelli. And the good thing about this model is it is actually 90% based on actual data. The model is based on mostly data directly from Dunkleosteus fossils. Barring that, it's based on data from other placoderms, and barring that, it's based on data from broader body shape patterns in fishes, especially those of similar ecological habits. And I would say that usually when I present this, I say this is part of a big manuscript being published in press, but the manuscript actually came out about a month ago, and it's now available free to read online, open access. So one thing you'll probably notice about this reconstruction is it results in Dunkleosteus that has a very short and deep body. I don't say short, it's not actually short. It's shorter compared to previous eel-looking reconstructions, but it's very deep-bodied and squat-looking. And this is actually this is a general phenomenon in arthrodires. And you can see this if you compare the three major groups of fishes that I use in this analysis relative to their body shapes. You have rayfin fishes who tend to have very deep narrow body shapes. This is a tuna, and tuna are actually unusually wide for rayfin fishes. Most are a lot skinnier. You have sharks, which this is very strange. Sharks actually have this very unusual stretched out body shape compared to other fishes. Like the body is very narrow, and from front to back, the landmarks are very stretched out, and it's not really clear why. But then they have a cylindrical body shape versus like a rayfin fish has a laterally compressed one. And then you have arthrodires, which they tend, with the same body weight, they tend to be a lot shorter and they tend to be a lot stockier. And a way to think about this would be rayfins, sharks, arthrodires. So, and again, this isn't just based on a bunch of random regression equations. Like, if you look at the complete fossils we have of arthrodires, they're all very squat-bodied animals. There's not much going on behind the end of the ventral shield. You can see this in like Cacosteus, Watsonosteus, and Cysoscutum. But even things like that aren't even arthrodires, even other placoderms like Naspis have not much going on past the end of the ventral armor. What's funny, though, is that despite these animals being really stocky, their fineness ratios aren't that much different from modern stocky-bodied fishes. So what fineness ratio is, it's basically a ratio of body depth relative to precaudal length. And it's a ratio of, I don't want to say streamlining, but it's kind of streamlining. And so all of these arthrodires plot out as below average among fishes when you plot out their fineness ratios, except for the chimeropisids, and they're weird. But they all plot out as being below average of the fineness ratio. But for example, Dunkleosteus plots out with similar fineness ratios to coelacanths, tunas, and groupers. Perciforms, a lot of that's groupers, especially because this is post-perciforms being split up. A lot of it's groupers. Another thing to consider is that a lot of people often think of Dunkleosteus as a very wide-bodied animal, and this is a lot because of the Cleveland Museum's reconstruction that you see everywhere. The thing is, this thing is reconstructed wrong. If you look at it, these side plates here are mostly replicas, and this was one of the earliest ones they did, and they didn't really know entirely how to put this thing back together. The later ones they had a much better idea because they had experience doing this. And in fact, if you look at the later reconstructions have a narrower body. They're not quite as laterally compressed as like a rayfin fish, but they're actually intermediate in body shape between a shark, where it's just like a circle in cross section, and something like a rayfin fish, where it's a bit wider. So they're a little bit laterally compressed. But Dunkleosteus stands out. The body is unusually deep, even compared to other arthrodires which we have complete body shape. And this is very apparent if you compare something like Dunkleosteus relative to something like Cacosteus. However, this looks like it's probably a factor of ecology. So this is actually a pattern you see in a lot of animals where a non-pelagic organism moves out into a pelagic adaptive zone. They tend to get these very squat bodies with shortened abdomens. You see this in arthrodires, but then you also see it in ichthyosaurs, sharks, and you see this in scombrids, which are tunas and their broader relatives. The ones that are like specialized open ocean species tend to be squatter bodied and have like, they're just squatter bodied, whereas the more coastal species tend to be more elongate. And again, this isn't just based on a bunch of regression equations. This is very evident if you actually just look at the fossils. So like, for example, if you look at these are various trunk armors of arthrodire species. Dunkleosteus has an unusually deep trunk armor relative to its length. Everything else is preserved as is. There's no evidence that the armor is actually shortened. It just looks like the dorsal ventral height has increased. It's just a deep-bodied animal. And this makes this thing really stand out in morphology compared to other members of this group. Yeah. And there's other features as well that kind of support this interpretation. There's an unusual apomorphic ridge on here that looks like it's a hypertrophied connection for the horizontal septum between the spine and the trunk armor, which is again similar to tunas and thuniform fishes. And the spine of Dunkleosteus, we have a partial anterior spine. It's really weird. it's got all these bony connections, and like the first 16 vertebrae are fused with the synarchal at the front of the skull. Like the entire spine up to here cannot bend at all because it's fused together. And this is kind of some of what you see in tuniform fishes, where the anterior spine is stiffened through various morphological means, and what this does is it provides a firmer base for the trunk muscles to push off of and basically make the tail fin go faster. And again, the shape of the trunk and the deep trunk mean this thing has very, very large axial muscles, especially laterally, the ones that would flex the trunk laterally. But again, most of the axial muscles are in an area where the trunk can't flex at all, which suggests something similar to thundiform taxa, where tendons are probably taking the force being generated by the trunk flexors and then just diverting it to the tail. So another way we can actually test these length estimates is we can use another method that's unrelated to the head, which you can use, let's say, the ventral shield. So we could take ventral shield length, and we could take the total armor length. And it turns out that these proportions are pretty consistent with total length in placoderms. There's a very consistent relationship between the two. Usually the ventral shield is about like 30% total length, and then the total armor is like 45%-ish total length. So we do that, and we apply it to Dunkleosteus. We get lengths that are pretty similar to what we get via estimations via OLL. So this is an entirely different metric that probably suggests that these shorter lengths are probably accurate. And this actually makes sense because recent discoveries of placoderms have preserved organ traces in the Gogo Formation of Australia. What they suggest is that the ventral shield and the trunk armor actually is analogous to the rib cage of a bony fish. They're not the same thing, but they perform a similar function that they enclose the organ cavity and they protect the guts from being damaged as the fish is swimming around if it hits something. And in these Gogo taxa, the anus is actually right at the end of the ventral shield, and the organ cavity just stops right there. And this basically agrees with what we see in other placoderms. The organ cavity just seems to stop at the end of the ventral shield, and it doesn't look like Dunkleosteus is any different in that regard. Another thing we can look at is the pelvic fin position. So in Arthrodira that we have complete pelvic fins for, the pelvic fin is almost invariably right at the end of the ventral shield. And this is what we kind of expect, again, if the ventral shield is covering the organ cavity and it ends right there. And what's funny is that pelvic fin position in fishes, except for some of these advanced teleos fishes that are weird and we don't really need to talk about, it's very consistent. It's a very consistent about 45 to 50% of total length. So if you can figure out where the pelvic fins are, you can make an educated guess on where the rest of the body is going. And we actually have pelvic fins for Dunkleosteus. They're very unusually small, and this is another feature that's probably suggesting ocean-going habits. And again, these pelvic fins are, hang on. So these pelvic fins were collection specimens that were collected off the cliffs of these major rivers. They couldn't go digging back into the cliffs several meters to get access to these bones. So I can't get access to the Cleveland Museum's records to actually tell where they found these fins. They're probably pretty close to the actual specimen, and that's the case for a lot of these. There's a couple of pelvic fins of Cleveland Arthrodira that are almost always closely associated with the body armor. So that suggests that you're not going to get like one of these things that's like a whale where the pelvic fins are way back in nowhere land, because you would never find them, because the spine doesn't preserve, and there's no way to actually track the body back into the rock to actually get to figure out what the pelvic fins are. And we actually have one specimen. It might show pelvic basals right in the armor, just where we see them in every other Arthrodira. The problem is that the specimen's not completely prepped out, and they look like pelvic fin basals. It's not 100% clear. And again, if you know what the pelvic fins are, we have a pretty good idea how long this thing is. It's the same length as everything else I've showed you. One final thing I like to talk about is I like to talk about the caudal fin shape, because that's something a lot of people are interested in with Dunkleosteus. So early reconstruction Dunkleosteus have this very ely-looking caudal fin shape, and fair enough suggests that's probably not accurate, because Dunkleosteus is an environment that's only inhabited by pelagic animals, and nothing that's pelagic actually has an anguilliform caudal fin. So they estimated it as having a more heterocercal caudal fin similar to a shark. And this was very well supported by the discovery of an Arthrodira with a lunate caudal fin, Amazictes, which was described about 2 years ago. So Dunkleosteus has a fin mostly following Farron's model, but there's a bit of it's a bit beefier compared to that. And the reason for that is that in most fishes, fineness ratio and caudal fin aspect ratio are very tightly correlated. In fact, you can see this in this picture here. In fact, if you look at the proportions of the trunk in Dunkleosteus, it actually would end up in this animal plotting out among laminid sharks and these pelagic ichthyosaurs. What's interesting is a lot of these old depictions of Arthrodires have these very ely-looking caudal fins as well. But when you go back and look at a lot of these specimens, one, the caudal fin is a lot shorter than these old depictions imply. In this depiction up here, it's a third total length, whereas in most of these specimens, it's maybe 20 to maybe as great as 25% total length. It's not that big. But there's also, in most of these specimens, there's a very clear caudal bend of about 20 degrees at the same spot every time in these specimens, except for some that have messed up, broken caudal fins, suggesting that even these may actually not have ely caudal fins. And there's precedent for this. This is a shark fossil from Monte Bocca in Italy. It's about 50 million years old. These are sharks. The genus is still alive today. You can look at that caudal fin. That caudal fin is very bent. It's very forked. You would not know that these sharks had a bent caudal fin based on the preserved fossils. If you just had the preserved fossils here and you didn't have the soft tissue outlines, because we don't have soft tissue outlines for Dunkleosteus' tail fin, or Crocodius' tail fin for that matter, you would assume that these things also had an ely lizard-like tail. What basically is the tail fin just gets bent along its longest axis when it's preserved because it's the path of least resistance. That's generally how this works. So scaling via the head and scaling via other plates of complete Arthrodires, it produces the same results you see here. You get 3 to 3.8 meters. What's funny is that these old lengths, these old studies that often claim, oh, we estimated and reconstructed the body based on Crocodius, their methods can't be replicated, and it's not even clear how they got those estimates in the first place. I suspect they may have used mandible length, but even that doesn't replicate their claims. It's just, I actually think there's some typos in some of these papers that led to people thinking this fish is a lot bigger than it actually is. Even scaling from Amazictes, which has an unusually small head relative to other Arthrodires, only produces a total length of Dunkleosteus of 4 meters. And there's no reason to think that Dunkleosteus was some super special exception compared to these other Arthrodires. It's following the same rules as every other member of its group. And again, what it looks like is that Arthrodires have a very consistent body plan, and everything is kind of in the same general place as, say, a shark or a bony fish. They just slap a layer of dermal armor over on top of it. Or rather, sharks and bony fishes have lost most of this dermal armor secondarily. So now that I've talked about this, we can actually try to estimate the size of the large specimen of Dunkleosteus. I've talked about CMH5768, but there's a couple of specimens that are even larger, and we can use this to get an idea of the maximum size of the species. This is specifically CMH5936. This is the large known specimen of Dunkleosteus. It is a partial lower jaw that has the front part with the bitey bits. And the thing is that this is by far one of the largest. There's a couple of specimens that are kind of similar size, but it's still abnormally large compared to the sample we have for this species. So putting this all together, we only get an estimated maximum length of Dunkleosteus of 4.1 meters. That's 13.5 feet. That's actually smaller than the largest great white sharks we have reliable evidence for today. So why does this matter? Why have I been ranting to you about the size of a dead fish for the past 45 minutes? Well, like I mentioned before, everything in biology is influenced by body size. Devonian paleobiology, and Arthrodira specifically, has lagged behind almost every other group of fossil vertebrates, mostly due to the fact that these things don't have reliable body sizes. There's tons of studies you just cannot apply to these things because you have no way to actually get an idea of how big they were. And, well, now we can actually study Arthrodira paleobiology in a much more rigorous fashion. And there's actually several studies hopefully building on this research that will be published in the coming years. Another thing is that Arthrodires often get this reputation of being slow, sluggish evolutionary dead ends. This is often due to the fact that they're very old, they're 306 million years old, and they have that dermal armor, which made people think that they're very sluggish, dull animals. This goes back to the very first description of Dunkleosteus, where Newberry has this blistering description of the whole thing of this group as just being a bunch of useless evolutionary dead ends. During this and other research has been suggesting these animals were much more active swimmers and occupied a very wide variety of niches. And this agrees with some previous suggestions of Arthrodires essentially being the Teleos of the Devonian in terms of diversity and the niche breadth that they occupied. This also suggests that aquatic ecosystems reached near modern levels of complexity by the end of the Devonian period, and this is pretty much in line with previous studies that have said pretty much the same thing. Again, another thing to think about is the Devonian has often been considered this very important period of vertebrate evolution because this is often considered to be the period in which vertebrates first got big. This is thought to be, traditionally, the Devonian has been seen as this period of explosive size increase in vertebrates, and it's largely driven by large taxa like Dunkleosteus. But again, this whole idea of an explosive increase of body sizes in vertebrates is largely based on patterns based on arthrodires. And as we've seen, a lot of these old estimates for arthrodires are essentially based on nothing. And when tried to be replicated, they're not as big. So basically, all of this is now in question. And what it suggests is probably a much slower increase to large size across the Devonian period, maybe possibly even starting as early as the late Silurian and a slow gradual increase to large sizes. It looks like these Cleveland shale arthrodires are just much smaller than people have been saying. These were thought to be, oh, these abnormally giant things. It's like, no, they're not that much different from other fishes of their time period. For anyone asks as well, yes, this does include Titanichthys and the other arthrodires from Cleveland shale. They all come out as being less than 5 meters, no matter what you throw at them. But probably the most important conclusion of this research is there is no strong evidence for vertebrates larger than 5 meters before the Carboniferous period. The largest arthrodires appear to top out at maybe 4.5 meters. Maybe they got to 5 meters if we're counting stuff like what we assume some could grow up to, and like we just haven't sampled them, but that's very speculative. We know how big the sharks got in the Devonian. The biggest shark we have is probably about 4.2 meters long. Again, less than 5 meters. The biggest other groups of fishes are, again, like 3.5 meters or less. They're not huge. This is actually a lot smaller than, for example, great white sharks. It's smaller than basking sharks, megamouths, whale sharks. And it's smaller than like 90% of whales. And this suggests that this idea of the oceans being filled with giant marine animals, which is something you see in modern oceans and you think about in the like extinct oceans, like you think about this with the Mesozoic oceans. This is probably a post-Devonian phenomenon, if anything. The first vertebrates that really seem to get to sizes much larger than than their ecological analogs in the ocean today are the Carboniferous, where we do get evidence of what's probably some 6-meter-ish edestoid sharks. And with that, I'd like to thank the various people who helped this research come to fruition, and then open the floor to any questions.
Can you go back about 2 or 3 slides? So, right there. You seem to take a broad leap from Dunkleosteus to arthrodires in general when you said that this idea of large body size in the Devonian was driven by arthrodires only, and you just kind of showed that they weren't that big, but really all you showed was Dunkleosteus wasn't that big. Now you did follow up by mentioning Titanichthys, but were there any other overestimated arthrodires?
All of them. Anything above 2 meters is probably not, because if you actually go back and you, I didn't show this here. Good example. There's this arthrodire called Liptaspis. It was recently claimed to be the largest known arthrodire, 8 meters long. The only known specimen of Liptaspis is a ventral shield that's this long. And the ventral shield, as mentioned before, is about 30% total length in arthrodires. That gives you a 2.5 meter animal.
So I know that you did show reconstructions for other arthrodires in your slides. I mean, right. I saw that. And what you're telling me is that that those were all, that people had overestimated all those as well.
Yes. Like, for example,
like I mean, like in the sense of a general trend. I mean, you mentioned you just mentioned that one example, but I mean, how sure? Okay, so there were a lot of. Well, you showed us early that there were a lot of people saying that Dunkleosteus was this great big thing. Did people actually say that about very many other arthrodires?
Yes, they said about Gorgonichthys. They said Dunkleosteus, Gorgonichthys, and Titanichthys are the 3 they generally say get huge sizes. Dinichthys is a 4th one that kind of they say it. The problem is all of these big things have similar proportions of their anatomy, and they look like they're all about the same size. Like, the biggest jaw of Dinichthys is about the size of an average sized Dunkleosteus. And like we have enough of these big ones to suggest that they're not like the basic proportions of their anatomy are not that much different. Like Gorgonichthys, that I'm doing stuff on Gorgonichthys right now, but Gorgonichthys is like about the same size range as Dunkleosteus. Titanichthys is about the same size range as Dunkleosteus. Titanichthys specifically, we have the sample size is not as good. The specimens don't preserve very well. But the odds of it being like 6 meters long are very low. Because we have decently preserved specimens. We have a couple decently preserved specimens, and they give you like 4 meters. In fact, this may be an overestimate because some of this is based on, some of these landmarks in Titanichthyes are based on landmarks that you'd expect to be abnormally large in a filter-feeding animal like a whale shark. And Titanichthyes is thought to be a filter-feeding animal like a whale shark.
So 15 years ago, I would always hear, oh, well, there's one other one that's probably as big as around the same size as Dunkleosteus.
That's Titanichthyes. Yeah.
I mean, when I would hear this kind of in passing, not from reading, but hearing one paleontologist or another kind of say this in passing, and it was always probably, were there actual reconstructions, or was that all just...
Gorgonichthyes is very poorly known. It's only known from probably about 12 specimens.
Well, Titanichthyes. Were there actually reconstructions?
No.
At all, or were people just saying that?
They were saying that.
Yeah.
I actually started this research trying to do Titanichthyes, and I was trying to use the old size estimates, and the paper completely fell apart because it turned out I couldn't actually make the mouth size stuff work reliably. I'm very frustrated with the fact that study didn't work out because it made a collaboration fall apart, and that was just very frustrating to not be able to make that work out.
Can you? This is probably me just not being able to mentally follow this, because when I saw your paper when it came out, all this made sense. But now I'm finding that I have a little mix up here. So when you show the Farron et al. 2017 reconstruction,
mm-hmm.
it looks in body shape pretty similar to
it does
to yours.
It does.
But then when you did your reconstruction to that same length, it was this long, skinny eel-like thing.
I know, because
did I hear you right when you said that the Farron et al. Reconstruction would require a larger head? Is that?
Yeah. So basically, I'm just getting.
But they know how big its head was. So how?
There's some stuff I can tell you it after this lecture. So I can tell you point blank, if you assume that length estimate is 7 meters long, that's what they claimed it was, and you take the known skull length of Dunkleosteus of that specimen, which you get something like that up there. If you try to estimate how long that skull is based on the reconstruction only, you get a skull that is about twice the size of the actual fossil.
So that just seems, it just seems really crazy, actually, to think that, that they made that reconstruction at 6.88 meters, but the only anatomical structure that is known isn't accurately represented.
Actually, all of them aren't accurately represented. They're all about twice the actual size on that reconstruction.
Huh.
In fact, again, you scale it to the actual size that I get, you get 3.7 meters, which is well within the range of error of what I get. You
mean if you take their drawing,
yes. and you
make that skull the size that it should be, and all the plates and everything, and then it turns out to be that size
yes.
of the bottom. Okay.
And what's really funny is that when that paper came out, no one criticized this. But when my thing came out and actually pointed this out, people got really, really upset.
Fascinating. Yeah.
Any other questions?
Do we have another class coming? Oh, you might have some questions on the chat.
Yeah, just checking. Yeah, it's recorded. Are there any other questions? Oops.
I was going to ask, and maybe you just answered that once you published this, was your work gaining traction or kind of revitalizing this whole look at Dunkleosteus that had laid dormant for so long?
I wish, but my impression is that this work is just getting ignored. That's my impression.
So it hasn't made, unintended, as big a splash as you would wish.
It did, but basically, I get the impression that a lot of the people who work on this group just kind of turn a blind eye to it because I'm not one of the people who work on this. It's one of those situations.
You're not part of the inner.
Yeah.
Circle.
Fish paleontology is funky.
Yeah, so it made a big splash in the news, but all the other paleontologists aren't on board.
It's not even they're not on board. It's that they don't actually have any evidence to argue against it, but they're not happy with it because they want a giant sea monster.
Well, it may not be a question of want. It may be a question of this was an accepted norm. And, once you step out of an accepted norm, there's normally, Dan and I were talking about a similar kind of situation on the way down. There's normally a tendency for a group to want to not accept,
Right.
anything that's outside of the norm. And, and sometimes it requires multiple voices
Right.
before that comes to a place where it's
So
adequately discussed.
that's true.
Perceive it as almost a threat to their core belief or something,
Yeah. So that's true, but I've heard some fish paleontologists say behind closed doors that yes, this length estimate is probably accurate, or at least much closer to accurate than previous ones.
I think your logic that you presented in assessing, the various body types and whatnot makes a tremendous amount of sense. And and I know from freshwater fishes that I deal with, you can see some very distinctive body characteristic shifts in certain things. I mean, if you see brook trout, for example, have got a huge mouth,
right,
proportioned to the rest of their body, or other fishes that are often that size. So, I mean, I don't think it's a wild stretch to think that phenomena existed,
right.
Over time.
I mean, that's the funny thing as well is I did all these head-body proportions. It turns out fish have a very narrow range of head-body proportions that unless you're specifically talking about like an eel or something that's eel-like, the head will almost invariably be between 1/4 and 1/6 total body length.
Mindy, do we have a class coming in here at 11 o'clock? I mean, do we need to be aware of getting out of here is what I'm saying.
This room is reserved until 11.
Okay.
I don't see any people
We should
crowd collecting outside
Yeah.
the doors.
Beating the door.
Do we have any comments on the chat or?
I don't see any. Does anyone have any questions?
Yeah.
Can I ask a couple just
Sure.
real simple?
Richard, did you have a question? I saw your.
Yeah, I just wondered how the Museum of Natural History.
Hang on. Your mic's not on.
Actually, it is.
Is there a way to unmute? I don't know if you're muted.
No, I'm not.
It looks like he's not muted, but for some reason we can't hear him. Maybe
Can you type it in the chat box?
The computer's volume isn't up.
I think it.
You there?
I think it is. Can you hear me?
Hello?
Hello?
Try now.
Hello?
Yeah, hello?
Yeah, can you hear me?
Yeah, I can hear you. Sorry.
Okay. I was wondering, how did the Cleveland Museum of Natural History take your data?
They weren't happy.
Please repeat the question.
Yeah.
Oh, they were wondering how the Cleveland Museum of Natural History took the results of my research. They disliked it. Does that answer your question?
Yes, it does. Thanks.
Pat, did you have a question? Your mic was on. I guess not. So here, how do we turn the sound off? You have more questions, Dr. Oldfield?
Well, these aren't really discussion. It's very short questions. I'm just curious about. You mentioned early on that there were 28 Cleveland Shire Arthrodires, which I knew that there were several, but I didn't realize there were that many. I'm curious to know how many of those are actually described.
Well, they're all described.
They're all described.
Except for one, they're all described. Except for Dunkleosteus, we don't know how any of them lived. Dunkleosteus and Titanichthys. There's Gorgonichthys, Holdenius, Stenosteus, Selenosteus. Titanichthys, Mylostoma, Paramylostoma, Bungartius, Hussicophia, which is very poorly preserved.
So they're all described as separate genera?
Yeah, there's a couple that are multi-genera species. The problem is that distinguishing between species of Arthrodires at this point is probably going to be very difficult. I've heard there's like Stenosteus probably has several species in it. I've heard suggestions. Supposedly Titanichthys does, but I'm pretty sure they're all synonyms. Like James Boyle, I think, suspects they're synonyms, and I'd go with what he says because he does the most Titanichthys work.
How many of those 28 were you able to include in your analyses?
None of them have complete bodies. But the thing is that I know what their bodies, like the size of their parts are. And again, if we go back to the whole, most like fishes, unless they're eels, have heads that are between 1/4 and 1/6 body length. The biggest head of anything in the Cleveland Shale is that Titanic specimen, and the head is 75 centimeters long on that. Like, to get a 5 to 6 meter Dunkleosteus, you need a head shield that's more than a meter long, which we do not have. Unless there's some magic head shield in Morocco that no one's talked about. And again, the thing is, like I said before, the stuff from Cleveland has never been in literature. There haven't been any published measurements of them. So no one knew how big this thing actually was. They could see it and they could say, it's big. The head on the big Cleveland specimen is 61 centimeters long. It's only about this long when you actually look at it. It's just very squat looking. They actually, at the old museum, they used to have it mounted in a foreshortened way where you couldn't actually get behind it and see how short the whole thing was.
I'm curious, where was that historical marker?
Which one?
The sign.
Oh, it's Sheffield Lake. It's west of here about an hour. There's this little tiny city park, which is supposedly where J. Terrell pulled out the first specimens of Dunkleosteus from the Cleveland Shale.
What's over there? French Creek? Is that where?
Black River, I
Creek
think.
is Sheffield. That's further in.
It's actually just on the coast. It's not a river. It's just on the coast. It's literally I've been there. It's literally just this little gully on the coast. And there's no river or anything. There are a few sites where I know there's this one site this guy has like. There's like this shelf that's extending out to Lake Erie, and you could like wade along it and you can see there's bits of Placodermi bones sticking out here and there. They've collected most of them, and they're at the Cleveland Museum, the Cincinnati Museum, but that's where they got them from. It's in the lake, but the lake isn't the sea. These things are in that lake formed by glacial activity 300 million years later. The sea is a saltwater inlet from the south, basically.
Incidentally, Peter Bunker came from Sheffield, which is, again, French Creek is a tributary of the Black River, and a few miles inland from the lake shore where.
I didn't know that. I just assumed that French Creek drained into the lake.
No, it's a tributary of the Black River. Yeah, Bunker, that was his home turf was Sheffield.
Yeah, Bunker collected a lot of this stuff.
Yeah. And that's one of the 100-year-ago paleontologists, right?
Yeah, I don't have. There's this famous picture of him rappelling down the Rocky River that you see a lot of, but I'm trying to see. I want to say one of those two people up there is Bungar. Actually, no, that's Bungar right there, the bald guy.
Yeah, I think
very interesting.
That's him, in the lower stout man.
And that's 5768 and 5936 right there. And those have been known for years and just never been described.
The other thing, Russell, is our mutual friend Mark Garland found a Dunkleosteus jaw in the upper Chagrin Shale in the Chagrin drainage. So, meaning that Dunkleosteus wasn't exclusive to the Cleveland Shale.
No, in fact, so let me go back to this. So, remember how I said that how the outer waters of the Cleveland Shale were very anoxic? The Chagrin Shale is just the coastal water equivalent of that, where where it was shallow, it was closer to the surface, and there was more turbidity, and the bottom was oxygenated. So you get lots of benthic fish there. You get not benthic fish. The Chagrin Shale's fish record is not very good. I have collected fish bone with Mark from the Cleveland Shale, from the Chagrin Shale. I don't know what it is. It's probably it's an Arthrodira, but I don't know what kind of Arthrodira. It's probably some kind of shallow water form, but it's the armor is very distinctive of shallow water form. But like they have these things called like reaper lobsters that are benthic animals. They have these things called phylocaries, which are benthic animals. They have brachiopods. So the Chagrin Shale has tons of evidence of animals living in it. It's got tons of evidence of bioturbation. You go to the Cleveland, there's nothing. There's no bioturbation. There's no benthos. There's no nothing. And the idea is that you can see this inlet here. The Cleveland Shale is the waters in the middle of the inlet, and the Chagrin Shale would be on the eastern side. And that would go all the way to hit the shoreline in Pennsylvania.
Wow. And that's a very fascinating map.
Yeah.
And that's just built up geologic.
Yeah. Like the shape of that, like there's a bunch of Devonian rock all along here. Like I know that there's some people who are working in like central Pennsylvania that are like right on the coastline where you have this freshwater saltwater interface and they're pulling out Devonian material. You go further east of that to like Red Hill, Pennsylvania, and it's all freshwater. It's all lobefin fishes. There are placoderms here, but they're all antiarchs. What's interesting is that they actually have found a few Dunkleosteus bones there, and they did some isotope testing on them because they would assume, okay, they're like sharks, they can't go in fresh water. No, placoderms don't care. Dunkleosteus goes where it wants. Dunkleosteus can go straight into fresh water, and it does not care. In fact, the reason sharks can't go into fresh water is because of their bizarre liver-centric physiology, and Dunkleosteus probably does not work that way. If you look at the arthrodires we have organs for, the liver is actually pretty small. It's about the size of a cod, so it's smaller than a shark, but it's bigger than a bony fish.
So when will you be presenting your dissertation?
Probably another year. I gotta get the data collected. The big issue is I work on mammals, and most of the mammal collections were not accessible for a long period of time because of COVID. So I have the data now, and I had issues getting the equipment, but I have the data and equipment now. So now it's basically a matter of sitting down and segmenting out surface scans of 575 mammal teeth and then getting them all measured is what's basically going on. So that's what I gotta get done.
Any other comments on the live?
If anyone has any comments, if you type them, I can see them a bit more easy.
Don't forget to record. That's what Rich says.
Yep.
I think we'll probably wrap things up.
Yeah.
Any other discussion points? Again, thank you both for helping put the technical side of things together. And Russell, was fascinating presentation. Again, this will be posted on our Ecophilia YouTube channel, which will be accessible from the Ecophilia website as well. And so hopefully this time next year you'll be famous with, millions of views. But thanks so very much, Dan. You got anything else, Dan?
Yeah, we can say we had them here. We had them.
Exactly.
But Russell, yeah, with all the work you're doing and preparation for your defending your dissertation, appreciate the time you spent on us this morning.